Okay, well, thanks for inviting me here today to talk about chlamydia STDs. I want to give you a little bit of background first. Chlamydia has been around for a very long time, since the 27th century BC. It's had plenty of time to evolve, but it's still evolving. It's a very prevalent organism, causes over 130 million cases worldwide each year, and those rates continue to increase. While we don't know what the true estimates are for trachoma, which is a chronic ocular disease caused by chlamydia, we estimate that there are about 250 million cases worldwide. And that's one of the reasons why the WHO has made chlamydia trachomatis a priority pathogen for vaccine development. There's been a lot of research on chlamydia, but it's extremely poorly understood. It's confined to humans. It is an obligate intercellular bacterium. You have to grow it in tissue culture over many days and sometimes weeks to grow it to sufficient isolation to freeze down. There are no ideal or limited animal models and no consistently reliable means to genetically manipulate the organism. This is the developmental cycle of chlamydia, and I only show this to point out that the organism has to be inside of a cell to form an inclusion to replicate, and that replication cycle is about 36 to 72 hours. Sometimes the organism persists, and sometimes it continues through its cycle. But this is what we're dealing with in terms of having sufficient genomic DNA for downstream analyses. So initial typing used morphology and tissue culture, including iodine staining. And this is just a microscopic picture of chlamydia-infected HeLa cells, and you can see how large these inclusions become. When poly and monoclonal antibodies were developed, they identified 15 serological types based on the major outer membrane protein of the organism. And that differentiated into three classes, or three seroclasses of the organism, B, C, and indeterminate. But these classes did not correlate with disease phenotypes. And once PCR became available, it was possible to take the gene that encodes for the major outer membrane protein and sequence it. It's about 1,200 base pairs long. And that differentiated the strains into 19 OMP genotypes listed here. And those genotypes correlated fairly well with disease associations. So here we have non-invasive urogenital strains that cause serositis, urethritis. Obviously, these infections can track upwards and cause upper genital tract infections, and they're caused by these strains here. The invasive strains are the lymphogranulovinarium strains listed here, and they can form an asymptomatic ulcer. The organism then tracks by lymphatics to inguinal lymph nodes and can cause what's called an inguinal syndrome. This can suppurate, and all sorts of fistulas can form. It can be quite painful and debilitating. And there are high rates of LGV infections in men who have sex with men. And also, infants that travel through an infected birth canal can develop ophthalmia neonatorum in the first month of life, and also pneumonitis within 6 months. And then there's trachoma, of course, but you can see that strains A, B, BA, and C cause trachoma, but some of these strains also cause urogenital tract infections. And with OMP genotyping, we couldn't differentiate what was going on there. So in comes MLST, and as you all know, it's a great tool, many advantages, especially for global epidemiologic applications, looking at drug resistance outbreaks, patterns of transmission, amenable to population genetics. You can study clonal diversification, estimates of recombination, and now there are over 120 curated organisms in the website, which is great. So what did we do with our scheme? Well, this was quite some time ago, and what we wanted to do was use genomes from a different species so we could develop an MLST that could be used not just for Chlamydia trachomatis, but also other species of the organism. But at the time, only under a dozen genomes that were available. But we used a blast score of ratio of greater than 0.5, and C. caviae, which is a guinea pig species, as the outgroup, and we identified 7 housekeeping genes that we could use for our MLST scheme. And they're listed here. This is a circular form of the genome, and I just include MOMP for your reference in terms of where that is in the genome. In our initial study, we used the 19 reference strains and then 68 clinical isolates from 6 different continents. And these resolved into 44 STs compared to 19 MOMP genotypes. There were identical STs that could be found in different countries for sexually transmitted infections. But STs for trachoma were restricted by country, and that makes sense because endemic communities are not mobile, and they're usually spreading infection within their own regional areas. In terms of phylogeny, the MLST resolved into 3 disease clusters. So there were the LGV cluster here, the non-invasive prevalent strains clustered here, and then the non-prevalent original strains here, and then the subcluster of the trachoma strains here. And with additional analyses, we could determine recombinant strains, and these are highlighted in green here. So we had a JA OMP-A. Of course, we used OMP-A as an initial reference, but that was not included in the MLST housekeeping gene profile. And so this JA is a non-prevalent group. You'd normally find it over here, but it was here with the prevalent strains. And the genomic backbone of this, or at the time, the MLST backbone, was of an E or an F strain. And we also found that there were neutral SNPs that were predictive of disease specificity. For example, these specific disease groups. So we did find that MLST can detect diverse and emerging Chlamydia trachomatis strains that, of course, we could use for epidemiologic and evolutionary studies of sexually transmitted infections and trachoma. So we did another study where we wanted to see if we could use MLST as a biological marker to detect C. trachomatis transmission between partners. And in each case, the partner had mutually identified the other as a coital partner within the last 30 days, and these were considered dyads. We used culture to make sure that we could grow up the strains and that they represented active infection. We did MLST genotyping, and we only used dyads where the MLST genotype was identical because, of course, our purpose was to see if MLST could determine whether or not these strains were identical or not. And of the 20 urogenital samples from the 10 dyads, each dyad had the exact same ST. So you can see here that when we mapped the MLST results to reference strain MLSTs, we got an E or an IA or an F. In some cases, we had SNPs, but they were identical between the partners. But in 4 of the cases, the MLST didn't match the MLST genotype we had. So that was interesting, and again, representative of recombination that's going on. So MLST is a valid subject-reported epidemiologic link between partners and can be used for partner tracing and evaluation of repeat infection, reinfection, or persistent infections. So we wanted to expand our MLST and include additional samples. So we used our 19 reference strains in addition to clinical samples that included a number from Russia as well as from Boston that hadn't been represented previously, and obviously additional samples from the Americas and Europe. This time, our samples resulted in 84 STs. Of course, we had a higher typing resolution in MOMP-A genotyping and a high discriminatory index. And our phylogeny was similar to our earlier phylogeny. And again, we found that we had SNPs that significantly associated with disease phenotype. And also what was interesting is we could differentiate ocular versus urogenital MLST strains, where the OMP genotypes looked to be ocular, but in the urogenital cases, the MLST was clearly urogenital, for example, a D, an E, or an F. So overall, the number of unique STs that we found were comparable across geographic locales. Russia had a significantly higher ST to sample ratio, which suggested a higher level of sexual mixing with introduction of alleles from other regions or reassortment of alleles. And we also found that almost 33% of our samples were recombinants, and the majority of these were from Amsterdam, suggesting selective pressure or adaptative diversification. But of course, the sample size is small. We wish that we had set thousands of samples that we could have looked at, but that's what we found from this particular data set. And of course, there are two other schemes. One was developed by Yvonne Panikuk, and she has been able to use her scheme to identify MLST for different species of chlamydia, and that's been very successful. And then Klimt et al. In Jorn Hillerman's group developed a scheme that did not include housekeeping genes, but 5 non-housekeeping target genes in OMP-A. And they've gone on to use that to type a couple thousand specimens using that non-traditional MLST scheme. So where are we headed? So one of the things we know is that MLST is still confined to reference laboratories. You need quite a bit of DNA for amplification and successful sequencing. For chlamydia, of course, oftentimes it requires culture to have sufficient genomic DNA, and you could obscure mixed strain infections because you end up growing up one over another. But you know, the analyses are getting easier with refined bioinformatic tools. And obviously it's still a powerful tool. So genomics and metagenomics, which we have been doing a lot of, gives better power for strain discrimination, more easily can discriminate the true trachoma from the urogenital strains that have these OMP genotypes. Of course, there's always improved technology. Now we're able to do whole genome sequencing directly from clinical samples without as much genomic DNA, and we use an agile and sure select probes that we've developed for this. And with metagenomics, sometimes the quality is not as good, and it obviously depends on the number of quality of reads that you have. But whole genome sequence is still extremely expensive, especially using direct genomic DNA from clinical samples using the Sure Select probes. It's about $300 per sample, but it does use less DNA than MLST. And the Oxford nanopore technology keeps getting better. The advantage is that you can do one sample at a time, whereas with Sure Select, you've really got a batch process. But you can get long reads. You can do sequencing from complex samples, and there's always improved analysis tools. But DNA sample quality and quantity may limit the precision of the strain typing in that respect. So I would like to acknowledge past and current members of my lab, as well as collaborators and my funding sources, and thank you for your attention.